I will move on to our next speaker, and this is our last speaker before we have a little bit more Q&A panel and break. Our next speaker is Dr. Ethan Perlstein, who is the CEO of Perlara, and he's going to talk about preclinical and clinical data from the past 5 years of using patient avatars to repurpose drugs for mitochondrial diseases like Leigh syndrome. So thank you, Dr. Perlstein.
Great to join everybody. Okay, good to see everybody here in the Mitoverse. I'm going to talk about what I call the Leigh syndrome cure odyssey. I'm representing work done here by my colleagues at Perlara, as well as our sponsor and collaborator, Cure Mito. Perlara is the world's first biotech PBC or public benefit corporation, whose mission is to end rare diseases. Over the last 5 years, working in the trenches with families, much like Richard has described, we have found a really great product market fit with mitochondrial diseases and inherited mitochondrial diseases more broadly. So I'll tell you about our experience with Leigh syndrome, and I'll reinforce a lot of the points that Richard made about defragmenting rare diseases, or bundling them together, despite perhaps their monogenic driver identities and despite other categorizations we might apply. Let's actually let the data decide how these patients cluster. And again, in a complementary fashion to what Richard has done, we exploit evolution as our hack here and take advantage of the fact that many of the metabolic processes that go awry in metabolic diseases can be modeled in very, very simple patient avatars like yeast. And so there'll be a little loss. Of course, that's a lossy model. You'll lose some of the predictive value, but you, I think, gain a lot of compression and versatility, because you can compress a lot of phenotype into a simple readout like growth, whether that's in a simple organism like yeast or something more complicated like a fly. So I'm going to summarize, and it's actually quite fitting that you mentioned ECHS1, Richard, because it comes up here. For the cognoscenti, there are many monogenic drivers of Leigh syndrome. It's many different types. SERP1 is obviously one subtype. ECHS1 is another subtype. And what we have found at Perlara is that we can build a bespoke patient avatar drug repurposing funnel that is, befitted for each of the genes that we're looking at. So for SERP1 on the left, we were able to exploit a yeast to fibroblast pipeline. And that's a pipeline we've also reproduced in other metabolic diseases. Alessandro talked earlier, you can go from yeast to organoids as well, but yeast to fibroblast is the sort of minimum viable pipeline. And on the right, for ECHS1, we used a fibroblast to fly pipeline. And what was pretty remarkable is that these independent pipelines were being pursued in parallel, and they independently arrived at the same compound, or at least the most promising compound from both of those pipelines was the same molecule, which I think is another example of how we can defragment rare diseases when we take this unbiased approach and just apply the best models. And sometimes the best model is, as easy as getting something from the patient's nose. And so this is a very complementary approach, but I think one that will end up also converging with what platforms like Unravel produce. So to summarize, I've given a talk at this forum now 3 years in a row, and some of this maybe you'll be familiar, but with yeast, we can, as I said, compress a lot of complexity into a very simple growth or no growth readout. And we've worked out a very streamlined process. It takes several weeks if everything runs without a hitch. And we can take yeasts that have various types of mutations, whether these are knockout mutations or whether we're modeling specific patient mutations. It doesn't really matter. The output is a sick yeast that doesn't grow, and then we can look for molecules that rescue growth. And here's a summary of a screen that we performed, and I'll just describe here the components of this process. So we have a SERP1 knockout. So in yeast, the SERP1 gene is encoded by a homolog called SHY1. That detail doesn't really matter, but we're working with a knockout mutant, and this is S. cerevisiae. And what you're seeing here is a plot of the results of a screen of compounds to look for molecules that rescue the growth of this SERP1 yeast mutant. And the blue points, the cyan points, the negative control, this represents the SERP1 mutant treated with a placebo. and each of those little dots represents an independent control. And you can see the black bar is the median. And then the purple dots and the positive control, the magenta dots, these are the positive control. These are the wild type yeast without any SURF1 mutation. This is what growth would normally look like. And this is growth under conditions where yeast are forced to respire. This is growth where they're given lactate as their carbon source. And the orange dots represent one of 8,400 compounds in a particular library that we screen a lot. And you can see those orange points that are floating above the cloud represent rescuers. And these are the compounds that we think are interesting. And as Richard pointed out, there could be these anti-therapeutic compounds on the opposite side of the distribution, the ones that are below the hovering or sort of sinking below the mass. Those are sensitizers, but we're obviously really focused on the rescuers. And we've already secured IP on this, so we can disclose the compounds. Of course, we're probably sharing this list with Richard and others. But we found with the SURF1 mutant, I'll just summarize that we found a relatively large number of hits. We've done almost 35 other screens like in this manner with other yeast mutants. And SURF1 is kind of special in that this screen yielded a lot of hits of different mechanistic classes. I've just put in bold and highlighted several that I want to focus on. Sertaconazole, we already heard earlier about the azole connection, and tolarazole. I won't go down that. I won't reiterate that point. Instead, I'll talk about some of the other molecules that came out of this SURF1 screen. It was a really rich, you can think of this SURF1 mutant as kind of this base camp. And there's many mechanisms that are revealed that are evolutionarily conserved. Potentially some are therapeutically relevant still in humans, we don't know. But this is this massive list here that we decided we needed to whittle down. So as a first pass, we took about a dozen of these compounds from the yeast screen and then tested them in a seahorse assay using SURF1 patient fibroblasts. And there are 3 compounds here that we are showing where you can see an increase in ATP production, and some are a little bit more dose responsive than others. But this was nice validation because of the dozen compounds that came out of the yeast screen, where all we knew is that they rescued growth of this yeast mutant and nothing else. It turned out that half of them could rescue or have an effect in the seahorse assay that would seem therapeutic. So that was very encouraging, right? The yeast model is lossy, right? You're losing, half of those molecules maybe are yeast specific, or maybe there's some trivial dosing issue. We don't quite know, but I think it's still quite encouraging that a very, very simple yeast model of growth and no growth of the disease can be predictive enough that it can then have a therapeutic impact in a functional measure of mitochondrial function in a disease cell. And that compound, one of those 3 compounds, compound number 1 is losmapimode. This is a P38 MAP kinase inhibitor. I won't go too much into the biology here, but just showing you some highlights of data looking at SURF1 patient lines. There's the ATP production effect, which of course is modest, but encouraging. And then we also saw what looked like effects on positive effects on mitophagy in at least one of 2 SURF1 patient fibroblasts. And that one responsive line was responsive in the ATP assay. So we already know from this that the fibroblast is not a perfect model, but this suggests that there could be some N of 1 selectivity to the responder status. But this was encouraging enough to think that maybe we could advance something from the P38 MAP kinase class into the clinic. And while this was happening with SURF1, in parallel, we were doing this pipeline for ECHS1 that involved a fibroblast screen first, and then validation in an in vivo model, in that case, a fly. And what we did in the ECHS1 screen is what we call a double metabolic challenge. It was really 2 assays in one. So we could take ECHS1 patient fibroblasts, we could grow them under conditions of galactose, which forces them to respire, and they don't grow as well. So there's simply fewer cells. You're looking here at these green specks are essentially cells. These are each individual cells, looking at a field of cells. And so the ECHS1 fibroblasts don't do well with metabolic challenge when you give them galactose, and they also don't do well when you give them propionate, which is a fatty acid. And those 2 stressful conditions result in less cell growth. And then we could perform a screen, just like we did in yeast, where instead of rescuing the growth of yeast, we're now going to rescue the growth of the patient fibroblasts under this metabolic duress. And that process led to a list of candidates, and this work was all done with Charles River, that led to a group of a kind of cohesive group of hits, all belonging to the same mechanistic class, P38 MAP kinase inhibitors. And that really, a light went off when we realized that this was a different form of Leigh syndrome. We used different disease models, and yet we sort of converged on what looks like a common rescue pathway. And to validate the work in the fibroblasts, we had an ECHS1 fly model tested by Clement Chow at University of Utah. And this in vivo model provided us even more conviction. And you can see here that when you knock down E6H1 in the fly, you get this small size larval phenotype. You can quantify that here on the right. And then treatment with a panel of P38 inhibitors, including losmapimod in the top of the four, resulted in rescue of two fly model defects or phenotype, disease phenotypes. Their size was rescued, and also their buoyancy. And so these two independent measures of physiological rescue in an animal, developing animal, really got us excited. And sort of the cherry on top, the coup de grace, was that another group independently of us, a company called Vyvan Therapeutics, they ran a fly lethality survival screen. So essentially doing exactly what we had done with yeast and with fibroblasts, but then actually did the screen rescuing fly lethality. And lo and behold, the top hit from their screen was losmapimod. So the totality of this evidence got us really excited about, could we have discovered that P38 MAP kinase inhibition is a convergent therapeutic pathway that could work across multiple Leigh syndrome disease drivers. And we don't know what the response rate will be per patient, but that's why you would go next into single patient studies. And so we've been, over the last few months, working with many moving parts. It's been actually much more complicated than we thought to run this study, because losmapimod was not something you could just freely get. It's not an approved drug. It's an experimental drug. It was actually a drug that was pursued by another company, and they failed in their phase 3. And so to get access to losmapimod involves negotiating with that company and a lot of other hoops to jump through. But we're really excited that with a pioneer ECHS1 family and a pioneer SIRF1 family, one in California, one in Texas, that we can initiate single patient studies for losmapimod in hopefully as soon as the next few weeks ahead. So this story tells you that it takes sometimes longer than you'd hope, but drug repurposing can result in very promising avenues. And it's not just hopefully going to be an endless string of N of ones, but we can defragment Leigh syndrome, maybe starting with this pathway. And of course, there are going to be plenty of other pathways. We've heard about the azoles and other mechanisms at play. And we know it's going to take a polypharmacological approach to really make a difference in SIRF1. So we hope that maybe losmapimod could be one pillar of a multi-pillar treatment stack. So thank you for your time.
Thank you, Dr. Perlstein. It's so interesting that such simple models as yeast and flies can really give us functional insights into how manipulating these pathways might be beneficial in people. And it's exciting to know that there are existing drugs to test in that direction.